All right, guys, I want to talk to you about some test taking tips for your AP Biology exam. Most of you guys are really well prepared. I want to make sure that you do as well as you possibly can, or as good as you possibly can. The first part of the test is multiple choice and math questions. You've got an hour and a half to answer together 63 multiple choice questions and then 6 grid-in math questions. The math questions are usually pretty easy. So I would recommend that you flip back and do those first. Make sure you get those points. Don't waste a lot of time if you happen to hit one that you don't know. But get the ones that you do know how to do, bubble those in, and then go back and work on the multiple choice. And again, I think you'll find most of those easy. Some, though, are really long, and one question may take up a whole page in the booklet. If you're running very low on time, I would skip those long single questions. Make sure you get as many answers as you can. Make sure that you wear a watch. Keep up with your time. If you see that there's just no way that you can get to some questions, make sure that you at least bubble in an answer. And what I would recommend is you bubble in the same answer on each one of those. I would say do B. For a long time, people taught C, and the test makers sort of know that. So sometimes they tend to avoid using C as a common answer. So again, if you just run out of time completely and you don't have any guess on something, use B. If you do have time, though, take your time, eliminate all the possible choices that aren't right, and then guess from the ones that are left. There is no guessing penalty, so make sure that every question on your test has an answer bubbled in. Section 2 of your test is your free response section. There's 8 total questions. 2 of them are long free response questions. You may hit a part or a couple of parts on each one of those that you don't know a whole lot about. But there are also going to be parts that you do know a lot about. The key to scoring well on this AP Biology exam is putting what you know on paper. Put every single detail. The assumption of the reader is that you don't know the answer. Prove to them that you do. Put as much on paper as you can. A long free response, a good answer for one of those should take probably a whole page or near a whole page. I would recommend you do them a section at a time. Usually they're going to be A, B, C, D. Write down A, answer part A. Write down B, answer part B. Write down C, and so on. Your answers do have to be in complete sentences. Don't use list or something like that. You can use a picture, but to get points, you must explain your picture. And also for you guys especially, make sure that you write neatly, and I would recommend that you print. There's also 6 short free response questions. The ones of those that I've seen have been pretty straightforward, but this is the first time they've given the real test, so we're not sure how they're going to look. You should know something about all of those. I think a good, thorough paragraph, 5 to 6 sentence paragraph, if you're direct and to the point, should be plenty to answer those questions. But again, make sure you put down everything you know and get all the possible points. To do this free response section, you're going to have an hour and 20 minutes to write. Plus, before you start, you're going to have a 10-minute period where they give you the questions and you have an opportunity to make outlines of what you're going to write. Use that time wisely. Write down key words and key phrases that you want to use in your essay. And then when you get the go-ahead to start your hour and 20 minutes, write all those things down. Doing well on the free response is the key to doing well on this test. Put everything you know down on paper. That being said, I want to do a couple of review points, and I'm going to start with evolution. I'm going to do a little bit with plants, and then finally a couple of genetics terms that I want to make sure you know. Remember, this is the first video in a series of 3. Please take the time to watch all of them on the weekend before the test. This evolution stuff, I think you guys are pretty well prepared on, but make sure you know the definition. Evolution is a change in the allele frequency of a population over time. Individuals cannot evolve. Groups evolve. The population is the biological unit of evolution. Another thing that I'll mention about evolution is that you can't pass on traits that you acquire during your lifetime. A guy named Lamarck had a theory of evolution based on the idea that acquired characteristics could be passed on to your offspring. This was the example we talked about of like a giraffe stretching its neck, and then its babies would then be born with a longer neck. Make sure you know that that doesn't make sense. You can't pass on acquired characteristics. Another thing that we want to mention is macroevolution, or sometimes it's called speciation. For one species, for one population of a species to evolve into separate species, it can only happen if you have two or more populations that are genetically isolated. That means they're separated somehow from each other, and that they're not sharing their genes. There's no gene flow between the two groups. That gene flow is cut off. And because they're separated, and because they're isolated from each other and gene flow is cut off, mutations accumulate in those two groups. And since mutations are random events, the mutations that happen in those two groups are different. So given enough separation and enough time, those groups may evolve into separate species. There's two patterns of speciation that I want to make sure you know really well. The first one is allopatric, and that word literally means different fatherlands or different places. This is when you have two populations of the same species that are separated by some kind of geographical barrier. It could be a river, it could just be distance, it could be a mountain. But because they're separated, and because there's no gene flow between those two populations, given enough time and enough mutations, they evolve into separate species. The second example is sympatric speciation. This word literally means same place. This is where you have a group of organisms of the same species living in the same place at the same time, but they're still separated somehow. It can be that they have different mating times. Maybe they have some kind of temporal separation. Their breeding times, or if you're a plant, your flowering times are different. So there's no gene flow between those that flower or breed at different times. It can also be due to polyploidy. And this is a plant thing. It doesn't usually work in animals. And this is where, through some strange breeding patterns, a plant gets extra chromosomes. And therefore it can't breed with the plants that it came from. That's called polyploidy. Two examples of sympatric speciation: temporal separation because of different breeding times, or separations due to chromosome number, polyploidy. All right, we also want to talk about once you have two separate species, how do species continue to be separate? The most common definition of a species is two groups of similar organisms, or again, let me start over on that. A species is a group of similar organisms that can interbreed with each other and that produce fertile offspring. But let's say we have two separate species, but maybe closely related. There are these things called reproductive oscillating mechanisms that keep them as separate species. So they can be prezygotic, which means they prevent the two organisms from actually making a zygote, a baby. Those prezygotic oscillating mechanisms can be temporal, meaning that they have different breeding times probably, so that they can't mate because they breed at different times. It can be mechanical, and just their reproductive organs, they don't fit, they don't work together. It can be gametic, that maybe they do mate, and their sperm and eggs get to each other, but when they get to each other, they don't work together. They can't produce a viable zygote. Another prezygotic mechanism can be habitat oscillation, that they just live in different places and they're not around each other. And then finally, can also be behavioral, that they have different maybe mating attraction behaviors, like a bird song or a fly pattern or a mating dance. And because they don't recognize the mating behaviors, they don't tend to mate with each other. There are also oscillating mechanisms that are called postzygotic, meaning that maybe the two groups are very much alike, and that given the opportunity, they'll breed, and they can actually make a zygote. Oftentimes, though, those zygotes, those mixed babies, the hybrids, are inviable, meaning they don't have the ability to live. They're inviable. It can also be like the situation with a mule, that the mule is born and it's very strong, but that it's sterile, so it can't make babies of its own. And then finally, we also get oftentimes in plants what's called hybrid breakdown, where a hybrid is produced, but after several generations, it either loses its ability to live or to reproduce. Again, these are reproductive oscillating mechanisms, things that keep one species separate from another. I also want to briefly mention some patterns or types of evolution. And these are fairly simple to understand, but the terminology is something you need to be able to know and explain. One pattern of evolution is convergent. You guys should know by now that converge means to come together. This is when two organisms that aren't closely related through evolution evolve to look very similar because they're living in very similar habitats, environments. Good examples of this would be like the shark and the dolphin, a fish and a mammal, or the sugar glider, which is a marsupial living over in Australia, and a flying squirrel, which is a placental mammal living in North America. Even though they're on different continents, the places that they live and the habitats they live in, the niches they occupy are very similar. So they've evolved to look very similar because that's what it takes to succeed in that particular niche or environment. The more common type of evolution that we talk about is divergent. And you guys know that diverge means to go apart, to become different. Divergent evolution is when you have a group of the same species of organism, you put populations in different places or different niches, and they evolve to become different species. A good example of divergent evolution is adaptive radiation. This is a key term to know. This is when you take a population of organisms of the same species, you put them into a new environment. And because of things like competition, they begin to evolve very quickly into different species. They begin to speciate. And they're doing that in order to avoid competition because there's some kind of limited resources like food or space. And it also allows them to move into new unoccupied niches in that particular environment. Some good examples of this would be Darwin's finches when they were placed onto the Galapagos Islands, and also the mammals that were on Earth right after the extinction of dinosaurs. There were all kinds of new niches available, and they evolved to fill them. They speciated to fill them. Another important type of evolution we want to talk about is coevolution. And this is when two different species of organisms evolve together. They help to shape each other's appearance, behavior, eating habits. A good example, or two good examples of coevolution are first of all, plants and their pollinators. Plants have evolved to have a specific color, a specific smell, a specific shape. And a lot of that has happened to attract and to lure in their pollinators. And on the flip side of that, the pollinators have a specific shaped beak or eating apparatus to fit that particular plant that they tend to visit. So that's a good example of coevolution. Another one would be like parasites and their host. Parasites have evolved different ways to get into the host, different ways to avoid the immune system. It's sort of like an arms race between the parasite and the host. We might also add up here predators and prey. They co-evolve together as well. If we happen to mention the word microevolution, that means there's small changes in a population, but not enough to cause speciation to happen. A good example of microevolution might be the peppered moth example that we talked about during the Industrial Revolution, or the emergence of antibiotic resistance in bacteria or pesticide resistance in different kinds of insects. It's small changes in a population, but not enough to cause them to become a new species. And then the last evolution topic I want to touch on is Hardy-Weinberg equilibrium, or sometimes it's called genetic equilibrium. We know that's a theoretical situation in which evolution is not happening, and therefore allele frequencies are not changing. To be in that situation, there are 5 requirements. First of all, there can't be any mutations, specifically mutations in the germ cells or gametes, because those are the mutations that are actually passed on and therefore affect evolution. There can't be any kind of immigration in or out of the group. Sometimes we call that gene float. We can't have gene float between different groups. There can be no natural selection or sexual selection. Another way of talking about the sexual selection thing is that the mating must be random. Mates aren't being chosen based on some kind of characteristic or trait. And then finally, you've got to have a large population. And that's mostly in order to avoid something called genetic drift, which is when allele frequencies change just due to chance. And that almost always happens in small, isolated groups. If you remember back in class, we talked about 2 types of genetic drift. One of them is called the bottleneck effect. And that's where you have a very large population that's wiped out by some kind of natural disaster, like a tsunami or something. where you only have a few survivors, and that the alleles of those survivors are not necessarily representative of the group that existed before the natural disaster. You lose a lot of genetic variation, and the allele frequencies change simply due to chance survival from a natural disaster. The other example of genetic drift is the founder effect. And this is a situation when you have a small isolated population, maybe like the Amish community. And then if there's a unique allele found in a couple of those original founders of the group, that allele may become very common in that isolated group if those people with the special allele happen to have a lot of extra babies, just by chance. So whenever we talk about genetic drift, remember that it's a change in allele frequency caused simply by chance. All right, topic 2 in this review video is plants. We want to talk about alternation of generations. And I've got a definition written up here. It's a plant reproductive cycle in which plants alternate between a haploid, sexually reproducing gametophyte, and a diploid, asexually reproducing sporophyte. Now that's a lot of words. So let's look at some pictures. Here's an example of how it works in bryophytes, mossy plants, non-vascular plants. Let's start with a spore. The spores are created by meiosis, so they're haploid spores. They're dispersed by the wind, and that's one of the reasons that moss are important in early parts of succession. They can come into place by wind. This type of moss is called the gametophyte. It is haploid, one end. There are separate male and female haploid gametophytes. Since they're already haploid, they can't do meiosis, so they have to create gametes by mitosis. The male plants do that in a place on the plant called the antheridium. They create sperm. And the females do it in a place called the archegonium. To reproduce, moss have to have standing water for the sperm to swim into that archegonium. And once they do, that creates a diploid zygote, which we see here. That zygote will grow through mitosis inside the archegonium and develop into a stalk-like structure, like that. That stalk-like structure is the diploid part of the moss life cycle, and it's called sporophyte, 2N. In the spore capsule, up at the top of the sporophyte, meiosis will occur. Meiosis will take diploid cells up there and produce haploid spores. Those will blow out in the wind and will start the cycle all over again. So the dominant part of a moss's life cycle is this haploid gametophyte part, and it's the part of the cycle that reproduces sexually. The shorter part of the moss life cycle is the diploid sporophyte part that asexually reproduces through the creation of spores. Ferns or pterophytes do something kind of similar. Let's start with the zygote. You've got a diploid zygote here, which will grow and eventually grow into a fiddlehead, which will unwind and become a fern frond. When we think of a fern, we are typically thinking of the diploid sporophyte version of a fern. On the underside of the fern fronds, there are these little sacs. They're called sori or sori. Inside those sacs, meiosis occurs. And meiosis will take cells from the diploid sporophyte, fern frond, and create, inside those spore sacs, haploid spores. So it's meiosis that's doing that, having the number of chromosomes. Those spores also will be distributed through the wind, will land on the ground, and grow into the haploid version of a moss, which again is called the gametophyte. It's one end. That gametophyte will produce sperm in the antheridium and eggs in the archegonium. And again, it's going to use mitosis to do that because it's already haploid. And once again, those sperm and eggs have to have standing water so they can get to each other. And once they do, we'll have a diploid zygote, which will start over. But again, moss are alternating between a diploid sporophyte that reproduces asexually using spores, and a haploid gametophyte, which is reproducing sexually through the use of gametes, sperm and eggs. And the gymnosperm cycle, like the pine tree or cedar tree cycle, is a little bit more like ours. Let's start over here with the diploid zygote. That can be found inside a seed. And since we're talking about gymnosperms, those are naked seeds. They don't have fruits. They do have little wings or propellers that help them to be dispersed from the parent. When we talk about a gymnosperm, we are talking about a diploid sporophyte, a 2N. Gymnosperms typically have two types of cones. They have pollen cones or male cones, and seed cones or female cones. In both of those cones, meiosis happens. And we know that meiosis takes a diploid germ cell and makes haploid gametes. And that's sort of what happens here. One diploid cell in the pollen cone creates 4 haploid cells called microspores. They're very tiny. They mature eventually into pollen grains, which are essentially containers for sperm with wings, because we know that gymnosperms are wind pollinated. In the seed cones, meiosis also happens, and it creates 4 cells called megaspores. 3 of those will usually die, sort of like polar bodies in human females. One of them will grow up to be what's called the megagametophyte, essentially an egg in a case. I'll take a time out there for just a second. All right, let me pick back up with meiosis created 4 megaspores that are haploid. 3 of those die, and we end up with 1 working female gamete, a megagametophyte. Now remember that gymnosperms are wind pollinated. So the pollen flies through the air, it lands on this seed cone, and once it gets there, the pollen grain is going to sprout a pollen tube, which you see happening right there. That's going to burrow down into the female cone and eventually find this megagametophyte. And once it does that, the sperm will swim down the tube, fertilize the egg inside, form a diploid zygote, and we start the life cycle all over again. So that's the general alternation of generations in a pine tree, very similar to our human version of reproduction. The last plant thing that I will mention is the way this works in angiosperms, flowering plants. When we talk about an angiosperm, we are talking about a diploid sporophyte. And remember that in angiosperms, the male structures are the stamen, and the female structures, which we see right here, are the pistils or carpels. In the anthers, which are the top of the stamens, meiosis happens. That also happens down in the ovaries of the pistils. In the anther, meiosis will create 4 haploid cells for every 1 germ cell that we start with. They're called microspores. These will mature into what are called microgametophytes, or what I'm going to call pollen grains. They have sperm inside. In the female and down in the ovary, 1 germ cell creates 4 megaspores. 3 of those die, just like in human females. We get 1 working, what's called an embryo sac. And inside that embryo sac are 2 important groups of cells. One of them is the egg or ovule that you see right there. It's near the opening or the micropyle. The other important cell is right in the middle. Sometimes it's called the central nuclei or polar nuclei. It's 1 cell with 2 haploid nuclei. Once a pollen grain is delivered by an insect or a bird or whatever kind of pollinator and lands on the stigma, it sprouts a pollen tube, which will burrow its way down through the style and into the ovary, eventually going up into the micropyle, into the embryo sac. Down that pollen tube will swim 2 sperm, 2 haploid sperm. One of those will fertilize the ovule to make a diploid zygote, and one will fertilize the cell in the middle, that central or polar nuclei, and make a triploid cell. The fertilized ovule becomes a zygote, and eventually a baby, an embryo. And the fertilized cell in the middle, the polar nuclei, that becomes the triploid endosperm, which stores food for the seed. So together, these structures that are circled become the seed. And the ovary that surrounds that embryo sac that usually swells up and becomes the fruit. And remember, the purpose of a fruit in an angiosperm, it's a good way to help distribute the seeds. So that's all of us about plants today. One more thing I want to look at. And these are just some genetic terms that we didn't spend much time on. One of them is polygenic inheritance. There are a lot of traits in people and in other things as well that aren't simply controlled by one set of alleles like Mendel thought. Things like skin color. If there was only one set of alleles that controlled skin color, we could have at most maybe 3 skin colors. We know that's not true. Height, human height, intelligence. Those are all traits that are controlled by multiple sets of genes. And anytime you have a trait that's polygenic and you graph like the occurrence of the different phenotypes, usually you end up with what's called a bell curve or a normal distribution. If we were talking about skin color, for example, we have just a few people that are the lightest light skin color. We have just a few people that are the darkest dark skin color. Most people fall somewhere here in the middle of our bell curve or a normal distribution. So if you ever see a bell curve of phenotypes, that's going to represent polygenic inheritance. Term 2 up here is pleiotropy. And this is when one gene or one set of genes has effects all over the body, multiple effects all over the body. An example of this might be cystic fibrosis. The actual genes that cause cystic fibrosis affect your ability to make mucus, cause it to be really thick and viscous. But since mucus is found not only in the respiratory tract, but also in the digestive tract and the urinary system and the reproductive tract, cystic fibrosis can cause problems in all those areas. Things like sterility, digestive problems, not to mention the breathing problems that are the main particular symptom of cystic fibrosis. Sickle cell anemia is also a good example. It's a problem with your hemoglobin, but it causes problems such as liver failure. It can cause problems with circulation. It can cause you to get infections. It has many effects all over the body. That's pleiotropy. And then the final thing, epistasis, is when a gene at one location or one locus affects the expressions of genes in other locations. And the best example I can think of here is albinism. It's possible that you could have sets of genes like this for skin color. And if you remember from class, the more dominant alleles you have for skin color, the darker your skin's supposed to be. So if you have all dominant skin color alleles, you should be really, really dark skinned. But essentially these alleles are telling your body how much melanin, how much skin pigment to make. But there's another set of genes at a different locus, a different location, that tells your body how to make melanin in the first place. Most people are homozygous dominant at that location. You know how to make melanin, and then these genes tell your body how much to make. But there are people that are homozygous recessive at this location. And that means their body doesn't know how to make melanin correctly. So it doesn't really matter what these genes say, because if you don't know how to make it, telling your body how much of it to make is sort of not so important. So if you don't know how to make melanin, you're an albino. You don't have any skin pigment, either hair pigment, eye pigment. That's a problem. But that's a good example of epistasis. And then the final thing is what's called hybrid vigor. You guys should know that a hybrid is a mix. And vigor means like strength. So this is saying that mixed organisms are stronger than homozygous individuals. A good example of this is, again, is sickle cell. If you remember, people that are homozygous dominant have normal hemoglobin. They don't have sickle cell anemia. They have normal shaped hemoglobin. People who are homozygous recessive actually have sickle cell anemia, and without treatment they will die at an early age. But people that are heterozygous for this trait, they don't get sickle cell anemia, but they also are somewhat resistant to a common infectious disease in Africa called malaria. So because they get that extra resistance to malaria, the heterozygous individuals are actually stronger for that particular African environment then are people who are homozygous dominant or homozygous recessive. So hybrid vigor. Hybrids are stronger than homozygous. And no sickle cell anemia is a good example. All right, this is the end of video number 1. Remember, this is going to be a 3-part series. Watch them before you take the tests.